mentorship.com, advancing life science research, connecting people and ideas.
Welcome to Mendel's Pod. Today our guest is Susan Molineaux. She's CEO of Calithera, a biotech startup in South San Francisco, and we're joining her in her office. Susan's gone from being a scientist at Merck to now leading her second company as CEO. She's become a mentor to Bay Area entrepreneurs and students. And last year she was selected as one of Fierce Biotech's 10 women of 2011.
Today's podcast is sponsored by AdGene, a non-profit that facilitates biomedical research with a library of published and useful plasmids. Join our collaborative community by depositing to the library or by requesting plasmids for your research. To date, over 1,200 laboratories have contributed to our collection of 18,500 plasmids. Find out more at www.adgene.org.
Thank you for having us over.
You're welcome. Happy to be here.
So what are you working on here at Calithera?
Calithera is a startup drug discovery oncology company. So we're working on novel cancer therapeutics. And we have a fully integrated preclinical team of scientists that are currently at the company and exploring, in particular, novel therapeutics for the area of tumor metabolism.
So how big's your team?
The company is 25 people, and 20 of those people are in the lab.
Okay. And how many products are you working on?
We are primarily working on one program. The target's not disclosed publicly, but it is a tumor metabolism target. We hope to have that fully developed and moving towards the clinic next year. And then we have exploratory programs behind that. The size of the team precludes us from working on multiple things at once, so we have a little bit more of a linear approach.
I'm curious because I want to talk to you about sort of the ideal model, your thoughts on that for drug development. And this is your second go-round with this, plus you were at Merck. Mm-hmm. So, we've had several guests lately talking about innovation in pharma, the model's broken, blah, blah, blah. And one of the trends is to go virtual. What are your thoughts on these virtual companies?
I think that virtual companies are okay in certain circumstances and are probably not the ideal model in other circumstances. The more you want the work to be novel, the more you need an integrated team with functional disciplines from various areas talking to each other to get to where you have to get, the more you can't use virtual in my mind. So we do work primarily in the lead optimization area. Everything we do is small molecules. So we have a big med-chem effort, a medicinal chemistry effort that goes on in concert with a biology effort. And those two groups have to be talking all the time, and we can't test a drug, for example, unless we can formulate it properly, unless we understand whether we're getting exposure in our animal models. And so those people have to come in and help with the biologists and the medicinal chemists. And we gain tremendous time efficiency by being able to turn the crank on one compound after another as quickly as possible. So in the old standard pharma model, you would have had all those people at least at the same site, if not in the same building, working together as a project team. And we've kept that model. It is an old model. And we think it's important for time efficiency. What's happened with the virtual model is it seems appealing now that companies such as ours have gone down to a pretty small size, that you could somehow outsource everything. Why do you have to have all those people in one building and pay them all? So the idea that seems so amazing is, don't we have clinical research organizations, CROs, all around the world that at various price points can do the work for you? And some people have tried to do this as a complete outsourcing exercise.
Yeah, like three-person companies.
Yeah, like the people inside the company just orchestrate, and the people outside the company all do the work. And without talking about the psychological implications of whether you have skin in the game or not, the issue that I see is that the people aren't in the same time zone, and they can't react quickly to the data, and most importantly, they can't discuss the data together. So I don't think the best ideas always come out. It works fine to outsource things when it's simple and linear, and it doesn't work when the biology is complicated, the work is complicated, and you have a lot of unknowns. So the more you innovate, the harder it is to run it as a virtual model. The reason it persists today is it still looks on paper potentially as cost effective. because the investors to biotech companies do worry about your burn on a monthly basis, and employees, of course, are burn. And they're permanent burn, in theory. So the idea was, just pay the CRO here, then pay a different CRO there, and somehow when you add it up on paper, it looks at equally or better in terms of cost effectiveness. But the question you have to ask is, at the end of running that for a couple of years, what data do you have? So if you don't get a product out the door, it wasn't really worth it to do it virtually. So I would say that the time efficiencies and the depth that you can go into as a group more than makes up for the seeming cost savings of doing it as a CRO. But having said that, our own project right now is very much in the in vivo efficacy stage, so we do a lot of animal work. And we wanted to test our compound in a variety of different cancer models. So we have some up and running inside, and then we have had to go outside to do some more complicated models.
Okay.
So that's an example of it's not worth it for us to develop all that technology, but we have control over the compound, how it was formulated, how it was distributed in the body. All of that we understand. So when we send our compound out to a CRO, we've already tested it extensively in-house. And I think there, the economy of scales are worth it. So it's a per situation. You can use CROs to your great advantage sometimes, but as an overall model, let's run biotech through CROs. I don't think it's going to yield as much as the more integrated non-virtual model.
And then you mentioned psychological implications.
Yeah, that's actually big. So after you get done having that kind of rational analysis, I don't know how you put a price on what it means to have a bunch of dedicated scientists with skin in the game in the room. I don't think you can put an economic price tag on that.
You brought some from your previous company, Proteolix, right?
Correct. I actually have people who've worked with me at 3 companies, so we're becoming like a family now. Mm-hmm. I was originally at Rigel when I came out to California, which is right down the block, and then at Proteolix, a company I started, and then here at Calithera. And there are at least 3 people I can think of have been at all 3 companies. So I met them at company number 1. We got along, we saw things the same way, and we kept picking up people who had similar philosophy. So after a while, you do have this enmeshed group that understands how each other thinks, and you know that you share the same approach to doing scientific research and drug discovery and development, and then you run even faster because you know each other. I don't know how you put a price on that one either.
So does this exemplify what you would consider your ideal model for drug development?
Yeah, that's a good question. I actually think the Calithera team, partly because I've worked with them before and they've worked with each other before, and partly because we've all kind of older, so the age group here is relatively more experienced. We don't have a lot of people coming right out of their postdoctoral training or their graduate training. So you have an experienced bunch of biotech people who've come from different walks of life, and they all get along philosophically. We've kind of chosen for that. We were able to do that partly because some of the management team has a following in the Bay Area, and partly because we got started 2 years ago when the biotech market has been at one of its low points, which means you have more people looking for jobs. So some really, really great people were either looking for work or were loose in the socket where they were. And you wouldn't find that so easily in times of really high biotech employment. So we put the perfect group together, and this is the most efficient team I've ever seen. On the side, we just worked out that we've done over 20 look-sees at various projects to kind of test them and reject them while we've been working on our major program. So we have enough efficiency to be able to explore and then, in this case, not work on the things that are less optimal. And that's important for innovation because if you're going to innovate, it's very hard to have insights on exactly what the right thing is to do. The whole point is you're in a super exploratory stage where people don't know a lot. So we have been able to kind of send out little pseudopods and explore things along the way while we continue walking in one direction. I think that's important. You need an efficient team to do it.
Okay, so not just put blinders on and pursue the one.
Exactly.
Because I was talking to a VC not very long ago, and he said what he was favoring was actually, to fund a team. And he thought the ideal, team was about 25 or so. Mm-hmm. But then he thought they could do 3 or 4 projects.
Right.
And that he wanted to fund the team more than the projects.
Yes, yeah. We're probably funded essentially more as a team than as a specific project. And I say that because the first thing where we brought something in from academics, unfortunately we weren't able to take it forward for a variety of reasons. And so we very quickly became a team with an open-ended approach that could look at a number of things. So that isn't ordinarily what would get funding. You have to have something concrete that you're working on to get funding. But we've persisted and again, been able to explore things on the side while we walk forward with a major program because the team is so efficient. And honestly, our investors are making at least a partial bet on the team, not just the specific project we're doing.
So if you did find an interesting project, side project, would you spin it off like, is usually done, right? The B, the C are spun out.
Right.
Or would you keep it here and pursue it?
It depends on the resources and where we are with project number 1, what we can do with project number 2. This team was formatted specifically to do 2 things, to be able to discover and lead optimize small molecules, which is a pretty standard biotech thing. But because in my previous company, we went all the way through late-stage clinical development, I also made sure we recruited and hired people who could do clinical development. So 25 people, and we probably have to grow to 30 or 35 to do clinical development. That's a pretty complete skill set, but nobody can, go out on leave or go on vacation. I mean, it's just the right number of people and no more. So we have very little excess. But we're able to move projects along. So if project number 1 moves into the clinic, we have plenty of capacity to do project 2 plus some exploratory things. So when you sketch it out, it's more of a linear thing. If you're going to work with a really minimal sized project team, which is very much what's in vogue with venture capitalists today, then the way you do multiple projects is in linear fashion. So if it takes 18 months, say, to get something from the discovery of a lead compound all the way through to moving it into development and it's on its way into the clinic, that chemistry, biology, biochemistry team can turn around and start the next project. If you're taking more than 18 months to get something to that stage, then there's something specifically interesting or problematic about the project. But ordinarily, if you were doing long-term planning, you'd say it's about 18 months on average to go from lead discovery to moving it into development.
So you mentioned that you started a couple years ago, and your Series A funding was $40 million, which is pretty good.
Yes.
I mean, today that's considered
Yes.
really nice. How tough was that?
Getting the $40 million was not that tough, surprisingly. But the reason for it is simple. First of all, a lot of my investors are repeat investors, so they knew me from before, and together we worked in a company where we were successful, and so we all benefited. So knowing the team is experienced and knowing the team can execute is definitely a factor when venture capitalists look at funding. The $40 million was partly due to the uncertain financial climate that we found ourselves in 2 years ago. It was very important for them to have investors around the table that could finance through a value inflection point. And the obvious value inflection point is to get far enough into the clinic to show that you have some interesting signal or some potential efficacy. And when you run the numbers, at least in cancer, 40 million is about the right number to start on something and get far enough into the clinic to get an answer on whether you've induced value or not. So you'll notice that there's a lot of $40 million financings that have happened in the last couple of years in oncology or in drug discovery. It's the new number that basically combines the old Series A plus Series B, and you do it as a flat round, potentially. However, it gets you to a value inflection point. So it's a way of ensuring that everybody's committed to go that far. Different than the old model when people felt that you could just get started. And once you had some interesting preclinical data, you might have a value inflection point there and be able to do Series B. But in these uncertain financial times, it's not so clear that you would get a jump in value, so you might as well just finance all the way through. So we're not unique, and the 40 million turned out to be unique maybe 2 years ago, but it's been replicated in multiple companies, and all of those companies are being financed to value inflection points for the same reason.
So we've mentioned your previous company a couple times, Proteolix. You were the CEO there. You founded the company, I guess. Mm-hmm. And then you sold it to Onyx for over $500 million. And just today you were telling me you had heard some news about the drug.
Right. Right.
Can you share?
Yeah, this is really unusual circumstances. So I played many roles at Proteolix. I was the founder. I was originally the chief scientific officer, but during that time actually ran the company with the help of an interim CEO who stayed on the East Coast and trained me to run things. And then I ran the company. And then in the last year, in 2009, a different CEO came in to position the company either for commercial launch of their product or a sale of the company or partnership or whatever. And I concentrated in the last year, I went back to being CSO and I ran the clinical and preclinical scientific side of the company. So I played many roles in the company. But being a founder and having been there the whole time and being the shepherd of all of the science, the main thing that we did was develop a compound, a drug for multiple myeloma. It's a proteasome inhibitor. It's an improvement over, we think, a current proteasome inhibitor out in the commercial world. So when we started the company, Velcade, which is a Millennium Takeda drug, had just been approved. It was a novel therapy, a proteasome inhibitor, and it was uncertain what its market value was going to be. In multiple myeloma, it turned out to be, I think it's $1.5 billion in sales a year now. It's been very, very successful therapy for myeloma. We took the risk that that might not be the case, and we also noticed some deficits with Velcade. Some of it was how potent it was, some of it was some of its side effects, and we worked to improve that. And we were very fortunate that we made the right guesses and we came up with a compound that had some virtues beyond Velcade. One of its virtues in the clinic was that it continued to work in patients who had already taken Velcade, even had some benefit from Velcade, but were now refractory to or resistant to Velcade.
What's the name of the Proteolix
So
drug?
the Proteolix drug is called cafilzomib. That's its generic name. It's now been commercially named Kyprolis with its acquiring company Onyx. In 2009, we had already run over 500 patients in the clinic, primarily in multiple myeloma. We had finished an accelerated approval single-arm trial. So we had generated all of the data that one needed from an efficacy standpoint to actually go to the FDA and seek an accelerated approval, which would be an early approval, and it's a very risky thing to do. When we were acquired by Onyx, all that work had been done. Onyx obviously picked the ball up and continued to do a lot of clinical trial work in addition to what we had done. But interestingly, last week, the compound went up before the FDA ODAC committee, the Oncology Advisory Committee, for a look at accelerated approval. The FDA will rule in another month whether it approves cafilzomib or not. But the ODAC meeting was quite exhilaratingly positive from my point of view, and they voted 11-0 in favor of the drug. And that was not a foregone conclusion. Onyx had to fight hard to point out to the FDA what all the reasons to give the drug to the public. Was it safe? Was it efficacious? All of that. It's hard to do an accelerated approval because it's really a limited amount of clinical data. And the FDA will only approve a drug at the accelerated approval stage if they feel that the benefits far outweigh the risks. And so it was incredibly gratifying to all of us who were at Proteolix to see the drug get such a positive response from the advisory committee. And the best thing about the ODAC, which I watched every minute of, was the last half hour of patients. They had a whole series of cafilzomib patients get up and talk, and it was obvious that the room went quiet. Everybody was really paying attention, and it was stirring to hear people talk about how they viewed as cancer patients what it meant to take a risk with a drug.
I mean, this is why you're in this, right?
Oh,
One of the
it's
biggest
the only
motivators.
reason we're in it.
Yeah.
That and some scientific, intellectual, satisfaction. But basically, there's nothing like seeing the patients' responses and hearing them talk about why they're willing to take some side effect risks or some toxicity risks to get a chance to take a drug that could prolong their life. Puts all this scientific debate that we go through about efficacy and risk into a different light. And it was really great to hear them talk. And yeah, I mean, if you talk about where's your satisfaction moment, doing a drug discovery development career, it's in listening to those patients talk. All the other stuff is secondary.
To what do you attribute your success?
Luck to some extent, negative persistence.
Negative persistence, what is that?
Negative persistence is what scientists have a lot of. They're optimistic in the face of many, well, it's skepticism, so they don't believe something unless it's real. But also, they are optimists that the next experiment they do, despite all the failure that you have in exploratory mode, the next experiment will hit. I mean, they don't give up with 10 failures in a row.
Okay, so the negative are the things that are always happening, and they still persist.
And they still persist. They come in the next day and do more experiments. The more innovative your work is, the more you're going to come up against failure.
And this company here, Calithera, is more innovative than
Calithera is even more innovative than Proteolix. We ran the risk at Proteolix that we could not have a drug that really had unique properties. So you were constantly worried and taking the risk that that might be the case. So when of course, negative persistence means you just keep going even though you haven't always succeeded. But I'm also comfortable taking risks. I'm very frustrated when things don't work, but in the end I come back and I'd rather do that than do something safe. That's really what it comes down to. So I would attribute my success to that. So it's personality, it's negative persistence and ability to take risks, or rather do that than safe. And definitely luck, being in the right place at the right time, and using my experiences to learn. I have no problem doing things that don't work as long as you're learning from them. So you have to be able to integrate everything you've done prior to what you're doing now and use that. Some people call it gut, but it's really your experiential knowledge.
When you say gut, you mean instinct?
Yeah, people say instinct because you can't articulate exactly why you're thinking the way you're thinking. But after 30 some odd years in this drug development world, I have some feel for the kind of risks you are taking or not taking, and what might work and what might not work.
Right. The gut is actually the combination, the amalgamation of all that experience. It
is.
Right?
It is. And everybody has a gut. The question is, how much are you using it? So I would say that I use that more than most people because I like to think about what mistakes I've made, and I have this optimistic view that you can always do it better. So I don't shun, looking at that kind of stuff, and we talk a lot about criticizing ourselves, being self-critical within the team. And not everybody I know does that. My first response when I was talking to one of my investors, he was congratulating me on this positive ODAC outcome, and he said, you did a great job or whatever, and I said, I just want to do it again. And that's my first response. I just want to do it again. So it's not about, resting on your laurels and sort of accruing prizes. It's because the drive is to do it and do it better. So Calithera comes out of a very successful set of programs at Proteolix. We actually had a second drug that went into the clinic from Proteolix. It's in the clinic with Onyx now. So we've had 2 successes potentially, and there is even a third drug that isn't in the clinic but could have been in the clinic. So that's an amazing amount of success. And because of that, I feel I can take more risk with Calithera because we've already succeeded. It's not that we need to succeed, it's that we want to keep pushing the envelope, and we're willing to take more risks to do something innovative. So the success keeps building to make you want to do it more.
Let's go back in your career and sort of trace maybe why you're this way. So when you were young, your dad gave you a microscope, and that was the start?
Yeah, so I come from a family that was very education-oriented. My parents were actually son and daughter of immigrants, and they felt that the education system in the United States was the pathway to success, but not so much financial success as that it was a tremendous benefit and privilege in the U.S. to be able to get an education. So education was held up as the great thing that you should want. Not for any means to an end, but just by itself. And with that came this feeling that you should learn a lot and you should explore. My father was an electrical engineer, so he loved all things scientific. And I and my sister, who's an economist, grew up with this idea that math and science was really great, was really fun, and there was certainly no gender barrier. Everybody should be thinking about this stuff because it's just so great. So growing up in that environment certainly helps if you have scientific and analytical interests to go in that direction. And being given a microscope as opposed to being told you have to get A's in school is a very different message. So if you're a parent, showing your enthusiasm for something and giving the kid an opportunity to explore is a really different experience than just, rewarding them for grades or achievements that are very linear.
And at the time, you were excited about the microscope. I mean,
well,
you didn't say, "Oh, what am I supposed to do?" Disappointment.
That was the test, right? My sister said, "Huh?" So she became an economist. What I liked about getting a microscope is the open-ended thing. I was just told, go play with it. So scientists really play with things. And I like just being able to put whatever under the microscope. So whether it was a little piece of onion skin, or at the moment I really remember is going down to the pond that was near our house and throwing some water on. And my father must have told me, go do that, it'll probably be interesting. And then you see things under there. So that kind of open-ended, unstructured exploration of the universe, and it's understandable, you just have to go out and figure it out.
And a connection with your surroundings there. I mean, it's not like you were in a class and the teacher said, here's a slide, and you look at the one
bacteria. Follow the instructions, right?
Yeah, but you're actually out there, looking at the pond water and connecting with the possibilities.
Absolutely, and let me tell you, if you want to be successful in graduate school in biology, I remember going into my first lab, and it's just like, here's your lab bench. You can work on anything you want, sort of, if you can convince me. It's so open-ended. I think it puts some people off to be so open-ended. They would like a little bit more structure. But the people who stay in graduate school and do research are all open-ended people, and they want the freedom to do things that nobody's done before. So back to the negative persistence. You have to want it, and then when you come up against things that don't work, you have to keep going. But I think that, you could hand a microscope to 10 kids and you could probably pick the scientists out by their reaction.
Nice little test.
Yeah, the little test. So I tried it on both my kids, and neither one of them is a scientist.
It went into the closet.
I didn't do that particular test, but.
Oh, okay. And actually, I heard you talking to some high school students, scientists, and talking about sort of career direction. And one of your points was stay sort of open and flexible, right? Because you started with you were a scientist. You went really, you were even thinking of the academic thing,
right.
And then you went into industry and you were Merck.
Right. Yeah, so there's two kinds of openness. So my original desire was to be a music major, and I actually stayed playing music through college. So I had a lot of other interests. I thought about being an English major. So I'm really open. I could have done any number of
English
things.
is really open.
Yeah,
that's
really
the most
open. So I'm not just a scientist, which partly explains why I got to be a CEO, because I'm not a pure scientist. So I don't think people necessarily keep the same academic paths, especially if they have a lot of interests. And then in terms of what you do with your education, when I finished my postdoc, I assumed I was going to go into academics. Other things happened and other opportunities intervened, and I wound up going to Merck, still thinking that I would do basic research. So I didn't step into my first job after my postdoc assuming I had switched over to industry and I was going to do something very different with my life. I saw Merck more as a basic research institute without having to do some of the grant writing and things you had to do in academics, and that appealed to me, and I liked working in teams, and that appealed to me. When I got to Merck, it was really probably 2 or 3 years later that I realized what I want to do is develop drugs, and I want to use research to do that.
How did you realize that?
I found myself being the only scientist, I was a bench scientist at Merck, seeking out the directors and going to lunch with them to hear about what happens after we finish the research, or what happens in a different department. And I realized my interests were very broad, and I got much more excited about the stories of people who had taken drugs into the clinic than the people who had just done really, really interesting basic research.
It reminds me of, I used to be in music, right? And most orchestral musicians, they just have their score,
right.
which is just their part,
right.
and they play through, and they never look actually at the conductor's
No,
score. This was a surprise to me when I was talking to them. I would
Yeah.
think they'd all know the bigger picture, but
Yeah. In music, I'm a little bit more the person who sat in the orchestra and didn't look at the scores.
So how did you make the decision go from the scientist to biotech management?
I didn't actually make that decision. The decision got made for me by other people to some extent. I didn't raise my hand and say, I just need to manage people. So my first big management job was a complete accident. It happened at Merck. Merck has a pretty hierarchical structure. As a bench scientist, I and many other people at Merck had no more than 2 research associates working for us, and we worked in giant flat teams. Unless you were going to go up and be a director, there would normally be no role for you as a manager, and it was the furthest thing from my mind. But a couple of years into Merck, we were working on a project that had a big spotlight on it. People up at the top were interested in it. And I was a molecular biologist at that time, and we needed a great deal of protein to be produced because we wanted to do a crystallography project. So we wanted to understand from a structural point of view what this particular protein looked like and how it looked bound to its compound. And all that means is you just have to make a boatload of protein. So it was kind of a simple job. But it wasn't working out. The team wasn't working. And the molecular biologists are the ones that control the bacteria that you grow up to produce the protein. So we're very far, as molecular biologists, you were very far from the end product, but you were at the beginning of the train. And a couple of other molecular biologists had been running the projects in a kind of, I'm at the beginning and I'm sort of gating kind of mentality. And I came in to fix the problem. I got kind of appointed to fix the problem after everybody was frustrated and angry at each other. And so I did intuitively what I do now as a leader. I just pulled everybody together and I said, well, look, we're just the people who produce the protein, first of all. All you people out there are the ones who need to work with it. So what do we have to do to get you everything and anything you want? Let's run parallel processes. We don't know which is going to work. And we just did a bunch of different things at once. But the main message was, we're all in this together, and I'm going to produce something that you need, and then it's going to work. So it's kind of parking your ego way outside the room and just saying, what does this project need to be successful? And I will do whatever it takes to make it successful. So you're the leader, at the same time you're in the trenches. And I was literally in the trenches. I mean, we grew protein up until it was coming out of our ears, and I sat there and spun pellets down with everybody. So it wasn't sit behind your desk and lead from behind your desk. But I found the process to be exhilarating, because then it was about going around to the various people, some of who were fairly frustrated with each other at that point because they'd been working together non-productively for a while, and trying to meet each person's needs, or trying to talk them into something that they weren't perhaps interested in doing, getting everybody's scientific input for what was a relatively simple project. But I decided I really liked doing that. And in the end, everybody was happier once I had taken over. And not because I was the leader up on a pedestal, but because the project team was more successful. It's a very different outcome.
And that was the source of the exhilaration for you?
For me, I was just happy getting the project team successful.
Yeah.
This was great.
So now, did you realize that you had this talent for leadership at that time?
At that time, I realized, wow, I did something other people can't do, or I can do it better than other people. And much more importantly, I really like doing it. And at that point, there was no chance at Merck to go any further up the hierarchy, because nobody would be picked out. The way Merck worked, you wouldn't be picked out for your ability to work with people effectively. You'd be picked out for your scientific, presence and what you had done to further the science of the project. And then after that, it's somewhat, I don't want to say political, but it's somewhat who you know and how you've cultivated other relationships. And I didn't want to stick around Merck and do that kind of thing for a while. So biotech was calling at that point, because the kind of skill set I was talking about, getting a kind of scrappy project team together and just getting it to work somehow, is actually more biotech and not the typical Merck model where appointed people come in and run things. So it made me also realize that perhaps organizationally, I belonged in biotech and not pharma. But that's just organizational model differences.
Yeah, when did you say, I'm going to found a drug discovery company?
Not for a long time after that. My first biotech company was just me sending out my resume along with my husband, who's a pharmacologist. We were both at Merck, and we actually sent out dual resumes, and somebody picked us up. It was a biotech company that needed both our skill sets. And that person brought me in as a vice president. So I went from a scientist at the bench to a vice president, which tells you something about title inflation in biotech. So I tried to grow into the role, because I certainly wasn't qualified as a VP at the time I became one. But that was somebody, again, saying, this is a person who is articulate. I can put them in front of the board. They can talk about the big picture as well as work with scientists, including some difficult personalities. It was pretty clear I could do all of that. So I think at that point my advancement was due to the fact that I like to look at the big picture and look at long-term strategy. So you say, good scientific skills, ability to work with people, want to work on a team, and then you couple that with being able to see the big picture and look into the future, and you wind up with a pretty nice set of leadership skills that you need for biotech in particular. I don't know if it makes you a great CEO of Procter & Gamble, but it definitely works in the biotech model.
For the CEO to be a scientist?
For the CEO to be a technical expert, so a scientist, for the CEO to like working in small teams that are very flexible and nimble, but being able to see the strategy as you stay flexible. In a larger organization, it's more about portfolio management and balancing, being able to write budgets and being able to articulate to different stakeholders what it is you're doing. So it's a kind of a communication and process job, and biotech is more about a just get something done job. Very different organizations.
Is there something over at least the last part of your career, where you're like, this is painfully obvious, and yet it seems to be, beyond people's ability to catch it?
That's a good question. So when I would bring things that I thought were painfully obvious up in environments like Merck, that's considered rebellion, and you're making waves.
Yeah.
And the big
So
organizations in general kind of are resistant.
Mm-hmm.
And so I think at Merck, for example, I had the reputation for being a rigorous scientist with a lot of enthusiasm, but perhaps somebody who was asking too many questions. So you go into biotech, that problem goes away.
Mm-hmm.
I don't
So
think...
that must have felt like a homecoming to you then.
Yeah,
Other people were doing the same thing.
I think you gravitate to the place where your personality allows you to be more successful. Biotech works for me. I could now predict what kind of people would do better in biotech and what kind of people would do better in a pharmaceutical organization, just by personality. But that's 30 years of being in the business and realizing, if you let the world take you where you belong and you don't fight it, you'll wind up in a place that's more suitable for your profile, whatever that is. So I don't think there's one set of universal leadership skills. What I'm saying is I think there are certain sets of leadership skills that work in certain environments. I'd probably be fine at making an independent film, for example. It's all the same problems. Mm-hmm. Do something with not enough money, not enough time, with a scrappy group of people who have strong personalities, but get it done and keep the vision. Sounds good to me. So I could probably make an independent film if I had film talent, which I don't have.
But that's where the film industry is going. I mean, more and more the big producers just fund these, right? They just buy them up.
Yes.
And some have compared that to the pharma model.
Yeah,
oh, you have?
It is, I'm one of the people who have, yes. That's been pointed out to me, and I agree. I think independent filmmaking coupled with big distribution channels is the same as biotech,
coupled with the pharma.
Yeah, and it's hard to come up with those models. But that's a case where if you want your film or your drug to reach global heights, you do need a structure and an organization that's very process-oriented that can do it. So biotech, if you allow biotech companies to grow into bigger companies, then they can become more process and organizationally oriented, and they can be successful. And that's the old model that produced companies like Gilead and Genentech here in the Bay Area. That model is very long-term evolution and isn't easy to fund right now, although I certainly hope that there are biotech companies that continue to grow into full-fledged companies. But it requires an organizational change and probably a few management changes along the way to grow into a fully, integrated company like a pharma company.
Mm-hmm. Mentorship's important to you.
Yes.
You say we should have mentors. And I think I read somewhere where you said men typically don't see the need for that as much as women.
Not as much. Not in my experience. Maybe the world is changing. I've heard lots of women raise their hand and say they need mentors, particularly women my age or a little bit younger, because it was a little more unusual for them to go into these careers 30 years ago, and so they looked around and said, hey, maybe I need a mentor when somebody else doesn't. But now it's evolved more into, who are you? Are you interested in being mentored or not? But I find men to be, by and large, not interested in that issue as much as women. There is no particular reason why one gender should be interested over another. Mm-hmm. If My experience is most people haven't had mentors. If you have a great mentor, I think anybody would then want another one. And the reason is because it's somebody who really supports you in your career development. That's my definition of a mentor. It's somebody who is looking at you very honestly, but is really cheerleading you all the way. It's not typical for a standard boss to be a mentor, although I think they should be. But the definition of a boss is more of an authority figure, which means they pass judgment on you and support you. A mentor is someone who's really just looking for you to find your right place and grow into your full potential. And a boss can't do just that normally. So I think you need a mentor. And I like mentoring people. Again, it's the same thrill as working on teams.
You're a mentor for, is it a student group here in the Bay Area?
Yeah, I sit on the board of a group called We Teach Science, run by Aragon Burlingame, who himself was an engineer and decided to leave the for-profit world and go for a not-for-profit organization that hooks up professional scientists as mentors to teach math, in this case, to students in middle school and high school, and do it remotely so that people can stay in their office. So that's a great way to solve the problem. But it's a one-on-one mentoring relationship between a science professional and a kid who doesn't necessarily have an interest in science. And in fact, this is mostly kids who are struggling. So that's hooking up those two groups. And then I myself have found myself mentoring a whole set of people in their 20s and early 30s who are doing various things in their career, not all scientists, and sort of the word spreads. So among my friends and colleagues, I've wound up with maybe 5 or 6 people that I've really followed their careers and made a change or had an impact on what they decided to do with their lives. And then there's my 2 daughters, which would probably say the same thing, that I've had an impact on what they've chosen to do because I will support them to grow to their potential as opposed to do some preconceived thing that everybody thinks is so great.
So today, a major decision from the Supreme Court on the health care bill. And so you're the lucky guest that we're talking to today. Do you have any thoughts on that?
I don't know from a drug development point of view what the real impact of that bill is going to be. I think it's going to have a big impact on American politics and thoughts about how people pay for things in the American economy. So it does talk to all this great health care that we have and that we've developed, who has the right to it, who's going to pay for other people to have the rights to it. But in terms of the global pharmaceutical market, the number of newly insured Americans that will have access to drugs, including expensive oncology drugs, is relatively minor compared to the global economy. The global economy used to be in pharmaceuticals primarily US and Europe and a little bit of Japan. World has changed dramatically with the rise of countries like China and South America and other Asian countries. The farm emerging, I think is what they're called now, countries make up a pretty sizable fraction of the global pharmaceutical world. And I think they're roughly, now they're about a third. So it used to be primarily US and Europe and maybe a third Japan, 2/3, 1/3. Now you have to make room for that 1/3 of emerging countries. That's going to change the way drugs are distributed and sold. It's going to change, who's going to pay for what. So it makes the U.S. less important, obviously, as a global market, it's a smaller piece of the global market. So I don't know how much impact that health insurance bill changes are going to have. It'll certainly have the benefit of having more care accessible to more people in the U.S. But the question of how that all rolls out and who pays for it has political consequences, obviously.
Thank you. Susan Molineaux of Calithera, she's CEO.
Today's podcast is sponsored by AdGene, a non-profit that facilitates biomedical research with a library of published and useful plasmids. Join our collaborative community by depositing to the library or by requesting plasmids for your research. To date, over 1,200 laboratories have contributed to our collection of 18,500 plasmids. Find out more at www.adgene.org.